All right, good afternoon, everyone. Let's make a start. So we've got two speakers this afternoon. The first one is Roberto Busi, and Roberto is going to be talking on herbicide resistance across the Australian continent. So looking forward to that. And then after that, we've got one of our PhD students, Hira, who's going to present after Roberto. So Roberto, after you. So.
So thank you, Ken, for the introduction, and to all of you that have come, or the thousands of people already connected remotely. I got asked to give a presentation. I was very happy to share the results of a paper recently published on the topic of herbicide resistance, and it was a good opportunity also to illustrate a bit what I am doing as a part of the School of Ag. So yes, I'm a weed scientist, as you can see there. We could see there. What are weeds? We start from really the ABC. So those are weeds. You will see those yellow wild radish flowering in a cereal crop. And in the next photo on the right, there are still yellow flowers, but that is actually the canola crop. And the weeds are just along here, the tractor track, the wheel track, and that is ryegrass. So two very damaging weeds that really they grow out of place. We don't want them there. Weeds can be very aggressive. You see here contrasting photo of a looping crop, one infested heavily with ryegrass and the other one where all the ryegrass is gone because of good technology, herbicide technology being used to get rid of those ryegrass plants. And so again, another example of radish being controlled effectively. Oops, it goes too far. Sorry guys, we got stuff in the beginning. Again, good morning, good afternoon. No, yeah, it's me that not very skilled. It's pointed. Anyway, radish being killed with a application of a herbicide mix. But surprise, surprise, sometimes, not matter how refined the technology is, how good the herbicide has been historically, this is a very successful herbicide developed by Bayer, and this was photo taken end of August in a trial up in Malwa, north of WA. It was actually a confirmation of something that we saw a year before here in the glass house. We moved to the field to confirm, and saw that with the leaf looks bleached, but the plant could survive. So many times this is what happens in the field after recurrent use of herbicides, and it's a problem. It's an increasing problem globally. The herbicide technology is more and more adopted, and therefore there is a steady increase of the herbicide resistance issue. In Australia, it's pretty much the same, and that is why there is a whole team named ARI. I took this slide from UWA, that is up in the up right-hand side corner. He leads the team to study herbicide resistance and try to mitigate the problem. I am part of the team, and I try to give a bit of a exemplification of the people that I've interacted more with. I really think that it starts with a who, because besides the topic of research, what really gets me excited is to collaborate with people that I enjoy to work with. So they are all summarized here, not that I have any enemies. Just don't get me wrong. I mean, you are very important, but I definitely do collaborate much more with Danica and Hugh. He is my supervisor, and whatever I characterize in the glass house on whole plants, and all the cows that I produce is passed along to Danica, that is putting everything in order and study at the fundamental level those herbicide resistance problems. I have Christine that from master students has now moved and healthy as a fixed term resistance research assistants. And then when we have something to say back to the growers or agronomists, we do a bit of an extension, and here is the extension specialist for WA, Peter Newman, and so I interact a lot with him. And Kit is a farmer that has retired, but there is quite a continuous feedback and interaction with him. So yeah, we try in our little own world to recreate a bit of industry stakeholders and try to see all the issues about agricultural weeds. Weeds can be pretty. It's just a little aside that is basically a plant that it can be pretty, but it just grows in the wrong spot or in a place that we believe is not where it should be. I'm not here trying to say that sometimes weeds are confounded as wildflowers, the vice chancellor did last year. This is a different topic. You go through Kings Park and you see very, very pretty flowers, but they are weeds anyway. So let alone the aside, let's go back to agri weeds. What I'm doing, this is my office in winter, is the plant growth facility at the back end of the campus where we grow most of the time ryegrass and white radish. And this is all the area that is full of pots, and we try to characterize and understand and quantify the problem of herbicide resistance. Personally, I've been funded for more than 15 years by GRDC. GRDC has heavily invested in ARI, and so it has been an absolutely privilege, and I felt all the pressure of this privilege, and so I tried to do more than what I am supposed to do. A little overview, then we will talk about the results of the study. But I thought it was really good opportunity to tell you exactly what I'm doing. Most of the time you see me running here, so you think that I'm running all the time. And you're right, I do run every day, at least once. But I also do experimental evolution, so I try to breed for herbicide resistance, and then sometimes the population that I use comes from fields, and they have already pre-screened in large screening sort of exercises, which is similar to what I will illustrate later on as the study that we just published. So each of the squares here host a field sample with seeds that come from different fields. And in this case, what I want to show is that quite a bit of emphasis in the study conducted was based on using herbicide mixture. So it's a multiple active ingredients used all at once and delivered to the weeds. And you don't need to be a scientist to see that the mixture works better than each of the active ingredients used as a standalone. But as easy as it can be to be seen how powerful they are, funnily enough, they very rarely have been tested formally and in a proper way. So that was a bit of the, let's call it, novelty that I brought to this study. And then, as I said, this will all be impossible to understand for you, but this is the simple model that we use to communicate the information back to the end users, to the agronomists or the growers. And we try to simplify as much as possible, but behind there are layers and layers of very important, but also very refined research that allows us to then really deliver to the growers what is the state of art, what is the up to and most refined understanding that we have of different herbicides and how they should be used. The research I do, I try to supervise students and create project opportunities for them. Some of them in these pictures have already completed their master project. Some of them are about to finish. Some of them have finished, and then they work with us within ARI. And some of them, like Rex, has just started, and we are still trying to draft his research proposal for a PhD. And again, it's good because it seems that all the industry, again, all the different stakeholders somehow keep participating and keep interacting, because Rex has full-time employment with New Farm. Still, he wants to do a PhD, so he's enrolled part-time, etc., etc. So it's really very dynamic. I try to be as dynamic as possible, and I also collaborate with other researchers. This is just an example of a tender that was put out by GRDC. I responded. I wrote the proposal, and I involved Rick Llewellyn, an ex-senior lecturer at UWA, that now has moved back to Adelaide and works for CSIRO. There are other researchers like Fiona Dempster, economist, that is currently part of the School of Ag. And so we delivered this. We are in the final 6 months of the project, funded by GRDC, and it was a national project. It was a good. It's good to collaborate or re-collaborate with people that are well known at UWA or spend significant time at UWA. And I have a couple of accounts. I also keep a bit of space on Twitter. I mean, I mostly use as on board the diary. I don't really want to become a slave of an algorithm, and I just use it to remind myself what I did and to post the most relevant sort of bits or tip of the iceberg of my research. But again, having a profile also then allows, once again, interaction, networking with the industry that is reading and commenting. And sometimes I ask questions if I have doubts, and I do read what other people are discussing. I quite enjoy. And this is the last bit of the description of my profile that I thought was all relevant. I really do like to interact with agronomists. I've been from this year part of an agronomy consultancy firm, SAgE, and I'm always learning how difficult is the reality back in the field, where the situation sometimes is very complex. Treatments are urgent. It's difficult to grow a crop. It's difficult to grow a healthy crop. And the photo on the right there, where you have the purple ryegrass plant showing herbicide symptoms and a healthy, I think, barley or wheat, well, one of the two. It's very much what we try to do. We try to grow a healthy crop, or allowing growers to grow crops and removing the weeds. And again, that was my photo to basically show the interaction that I also have with chemical companies, and I really like to share or to become aware of the IP that they have and they share with me under agreement. Sometimes it's a consultancy that is all within the university framework. And again, it's good because it allows me to see where the industry is going in terms of herbicides development or crop protection, and I can sort of reshape or prioritize certain areas of the research that I'm doing, all focusing on weeds. So back to the study that I wanted to present very briefly. You will see the title that is identical to the title of my talk, and a long list of authors. Again, for the first time, we wanted to bring all of these agronomists, influential agronomists. Some of them are from WA, some of them are from South Australia, New South Wales, or Victoria. We wanted them to really contribute to the paper. They did initially because they collected from different properties with seed samples and sent them to UWA. I did this whole screening, and I provided this research service. I provided them with the characterization of the profile of resistance. But then when I wrote the final paper, they also contributed in the discussion, and we made sure that we were aligned with the final message that was going out at the very maximum level of what I actually do in terms of publishing in a peer-reviewed international journal. And I was really happy to have this collaboration from A to Z, from the beginning to the end, up to the formal peer-reviewed publication. So the study was a fairly large study. We received about 600 samples of wheat seeds running ryegrass, and I sprayed several herbicides, about 20. So again, I tested millions of seeds. So it was a very large database requiring quite a bit of statistics, and that is why one of the authors is a statistician, quite well known in the field of weed research. This is the full list of herbicides. I don't want you to go through, but I just want to say that yeah, the whole study was designed to understand resistance to this new herbicide that was the herbicide that was developed by BASF. And there was an agreement, about a funding from BASF towards the UWA of 5-digit type of funding, so substantial funding, and that enabled us to run the service, have the ability to hire people to do it, and then give the data to BASF, and then give the service to the many agronomists and growers that submit the samples. And again, the novelty was this proportion of the study that when we tried to publish in a peer review final paper, it was well evaluated, the fact that we used herbicide mixtures compared to standalone herbicides. And a bit of the methodology. So yeah, this is how one sample looks like after being prepared. We count 100 seeds, we put in a small square, say 5 centimeter by 5 centimeter, and then we apply the herbicide directly onto the seed, and then immediately after we cover, and then we gently water, and then we wait for 3 to 4 weeks. Sometimes we apply the herbicide on plants that are at optimal stage. You see how green they look, perfectly receptive for a herbicide treatment. And yeah, we wait 4 weeks, and then everything starts to look like this. So with weeds dying, but some of them are not. So this is a clear example of resistance. So something that we can't kill at a dose that was used or would be used in the field. And so then we quantify, and we simply count. It's a binary response, 0 dead, 1 alive. Sometimes the herbicide is applied onto the seed, and we see these little bushes of ryegrass emerging. And again, each tray contains 20 different samples from different parts of Australia. And so we quantify how many plants there are, and then we provide the result back to the agronomist. And sometimes some herbicide or herbicide mixtures are much more powerful, and there is not much resistance to report. And again, that gives us a clear view of and ranking of what works more effectively or less effectively. And so, yeah, we move into a bit of data analysis. We categorize. I'm sure that everybody knows how to interpret a traffic light. If it is red, likely it's a bit of a problem. Yellow, be aware of it. It's a bit of a bell that goes off, and if it's green, yeah, you're good to go. So same we did with our data, and help that massively the statistical analysis. And I don't want to share much because, I mean, the time is limited, 20 minutes are almost up. I just wanted to give you a general description of the ability of ryegrass to resist, to survive when we apply herbicide. So ryegrass was collected across the continent, across Australia, and just stop yourself to this 0. So it means that only 10% of the samples received had 0 resistance. So they were susceptible to all the 21 herbicide options used when they were used as a one treatment, focusing on all the 21 we used. But if we analyze the same sort of data set, but we restrict the analysis to the herbicide mixtures that we use, we see that ryegrasses are much higher proportion of ryegrass remain susceptible. So it was just, again, to re-emphasize that mixtures are much more powerful and allow us in certain conditions where they can be afforded, where there is little crop effect. Anytime they can be used, as a management tool for herbicide resistance, they most consistently deliver greater efficacy. And then, yeah, just 3 final slides to sort of show you how we try to analyze the data and give you a bit of a spatial distribution of resistance. So Roundup, everybody would have heard of Roundup, has been banned apparently in Germany a couple of days ago. Good luck to them. But it's been a very successful and the most adopted pesticide ever. And of course, ryegrass was able to evolve resistance to Roundup, but resistance is different in different states. And we clearly identified a hotspot for Roundup Ready resistance in New South Wales, and we reported that. And of course, I mean, we discussed that with all the pros and cons, the numbers behind that 40% versus all the other states. And so in WA is still relatively low. And again, this will be very useful for somebody like GRDC that wants to invest in the future in terms of prioritizing herbicide resistance research. Another example, it won't mean much to you. Maybe it means that you know, cherry trees flowering in Japan. It's a very successful herbicide, been used for 10 years. It changed dramatically the business of Bayer because apparently about 40 to 50% of growers every year use this herbicide. So it's a herbicide that is soil applied and control ryegrass very well in wheat. But we found, very worrying levels of resistance in South Australia and Victoria, and we discussed the why not. This was so different, for example, to WA, which was only 8%. And new herbicide, brand new launch in 2020. In some conditions, there was no resistance. So this is all I have to say. I hope you are still reasonably alive. And thank you for your attention. I just want to finally conclude with 3 sort of summary points. Again, the service that we have provided and we provide will continue. My research will continue. Currently, I have a fixed term contract to 2025. So the whole vision is to close the gap between what we see here in control conditions and what happens in the field. And it's out of order. But the second point was that this is a slight change that was not audited perfectly. Sorry about that. So we keep the collaboration keeps on going in terms of advancing the fundamental knowledge of what is happening. We have a new recruit of Ari, Candy is here, so she may also be involved. Candy, beware. One day I will come to your office and talk to you. And we are also moving more and more to the field to really validate our results into the field, and that provide us better opportunity for extending the message of trying to manage weeds, trying to mitigate herbicide resistance, and we collaborate with university and chemical companies. Thank you for your attention. Thank you, School of Agriculture. It's awesome to work within the school, and all to Hira, unless there is any question.
So do we have any questions? Thanks very much. Any questions?
I'll plant one. So Roberto, are growers successful in managing their resistant weeds, do you think?
Yeah, definitely they are. Very rarely when you try to, even I was trying to find an example of failure in the field, to show here how it looks like when we have resistance in the field. And very rarely we see large areas where weeds have not been controlled. So I've been to many fields, and by a combination of the fact that the crop is this year especially has been very healthy, so it's competed a lot, it's very difficult to see those type of scenarios. I mean, the one that I showed was actually an experimental type of field where on purpose those weeds were planted. and try to establish as much as possible. So then it would have been a very good site to conduct research in the field. So yeah, definitely my perception is that growers are investing heavily in crop protection, and they are quite successful because of the continuous pressure applied to weeds and the layers of complexity in terms of control. The strategies, control strategies, continuously applied from the sowing to the harvest.
On a trip up north or 2 weeks ago, it was evident that weed control is fabulous everywhere. I've never seen such good weed control in northern crops. So the radish is under control, it seems. There's a predominance of Roundup Ready canola use. Would you like to comment on the use of Roundup Ready and how it may help or hinder ryegrass glyphosate tolerance?
Yeah, these are the results from 2020, and for WA, what we found in ryegrass, one species only, that is the most important. I mean, there is also radish that, and definitely the Roundup Ready canola technology has helped to keep radish under control. But I did the same study restricted to WA in 2021, and the 3% has become 13% in 2021. Ryegrass, Roundup Ready resistant. So when you have 2 points, you can draw a line through them. I don't know whether that is already a trend or just fluctuation between years. It seems that the theory would say that resistance remains rare, and we all know that is 1 in a billion of it, especially for a successful herbicide as Roundup being used for many years. Still, the level of resistance is low, but somehow it is also low because we have been good probably to control and keep the population low. So then the problem does not explode or is not seen when you then drive through paddocks and you don't see this explosion of failure in weed control. But it's definitely there, and the perception also within the agronomists is that the threshold of the initial frequency of resistance is not 1 in a billion, it's probably 1 in 1,000. And that is why somehow, not that we all of a sudden have to rely with 20 herbicides applied every year. There is also a cost to do all these operations. But it definitely seems that it's an issue that there has to be some investment in thinking and trying to manage or to reduce.
Yeah, I have a question. And I see in America, they will use the laser to kill the weeds, and the card with the AI system also can distinguish which is the crop, which is the weeds. But this method can replace the herbicide.
Yeah, definitely. There is a push to develop new technologies. You mentioned 2. One is like zapping weeds with laser or electricity or high pressure vapor, and then the other one is try to do it maybe when the crop is present. So the crop is green, the technology is called green on green. So the technology is also under development here, and with our ability to deal with big data, it's definitely something that in the future may lead to an optimization of what we currently do. Probably won't be anything that is very disruptive, meaning changing dramatically, but definitely that will lead to a good optimization, and maybe again trying to reduce the cost and be able to optimize the energy or the money spent on protecting the crop.
Thank you very much, Roberto. You mentioned that you're primarily working with ryegrass and wild radish. What is the occurrence of other species becoming what weeds in these fields? Do you have areas that are prone to that, or do you see it's quite?
Well, there are a range of locally important weeds. Ryegrass and radish have been our favorite, or have always been more important than others because of their greater ability to evolve resistance to herbicides. So it was also easier. It's easier for us to study them because they keep giving us or gifts, depends what type of bit of a conflict of interest. Yeah, definitely there are other weed species that are always in the emerging state, and sometimes they create locally some problems. You know, you have heard of matricaria in Meriden is a big problem, or barley grass or brome grass or cape wheat, depending on what system you've got. If you are a continuous cropper, maybe cape wheat is not much of a problem, but if you have sheep and pastures, and cape wheat, it's a problem, and it's difficult to control, and it's just enough, it's just sufficient to drive around to see how much there is, or how much there is on the side of the road. And so, yeah, definitely it's a continuous, sometimes if you drive and you see how much wheat there is on the side of the road, it seems a bit that we will never get. I mean, actually, we are pretty sure that we will never get rid of them. But
you mentioned the survey that was done by Michelle last year.
Yeah. Michelle has been every 5 years going out and collecting a range of wheat species, including also wild oats. So. Yeah, even in different parts of Australia, some weeds are more important, but not in Queensland, you know, ryegrass is not an issue at all, although since it is also moving into summer. So yeah, weeds keep adapting. They won't be eradicated, and maybe every now and then there will be one that from emerging becomes emerged and becomes very important. So there is a new publication from.
I think one of the biggest challenges that we talk about is reducing herbicide use. One of my PhD students published a paper showing that on average, I think it was about 6.3 applications of herbicide on every paddock every year. So it shows you how much herbicide is used, and that's one of the challenges we've got the resistance folks to use non-herbicide methods to control. So thanks very much for your I'd like to introduce our next speaker. It's my pleasure to introduce Hira. Hira is one of mine and Matthias's PhD students, and Hira is heading towards the end of her PhD. So she's just got one paper accepted, which is great. Well done, Hira. And has another couple of papers and trying to finish up her chapters. So Hira is going to talk on the use of electromagnetic induction for leptospermum, for measuring soils and leptospermum survival.
Yeah.
So Hira, over to you.
Thanks so much. Thank you everyone for being here. I will be discussing about my ongoing project of soil mapping using electromagnetic induction for Leptospermum nitens survival and growth in Western Australia. This project is a part of our CRC honeybee program, and this is generously funded by them. So Leptospermum is a plant from Miteraceae family. It has 300 node genera, and it has more than 3,800 identified species. This plant is a native Australian species, and currently there are 85 identified Leptospermum species in Australia. And out of these 85 identified species, 15 of them are only found in Western Australia. These plants are highly adapted to a wide range of soil conditions and environmental conditions naturally. And they are popular for their essential oils, ornamental shrubs, and the quality of honey. So what is Leptospermum honey and Manuka honey? Leptospermum honey is the honey produced by the bees when they feed on the nectar produced by the Leptospermum plants. So this honey has a medicinal value because of the presence of methyl glyoxyl or dihydroxyacetone chemicals. Because these chemicals act synergistic to common antifungal and antibacterial agents. And these things make it different from a normal honey and increases their value. Currently, Australian honey industry is worth over 100 million, and there is a potential of increasing this return by increasing the production of Leptospermum honey. So CRC Honeybee Project 6 is designed to investigate the performance of Western Australia native species, Leptospermum species, in different soil types for their optimum survival and growth, which will ultimately lead to flowering and nectar yields, and for high value honey production. So this project, the key actions for this project is to map the soil for commercial Leptospermum plantations, and then correlating those soil maps with the plant growth rates and the plant survival in the field, and ultimately correlating those soil maps with the quality and yield of nectar produced by these plants. This current research only deals with the first two steps of this project. So two study sites were selected for this purpose. One is Bajingara. This site has a uniform sandy texture soil, and this is approximately 16 hectares of area. And the second site was in Kukring. This is a site with a variable texture soil, and this is approximately like 20 hectares area. Soil mapping using electrical conductivity sensing. For soil mapping, we use the electromagnetic induction sensor, 𝑬𝒏𝒗𝒊𝒓𝒐𝒏𝒎𝒄𝒊𝒆𝒏 we use this sensor because it needs the minimum contact with the soil, and this makes it mobile. So we put this sensor in a non-conductive sled to make it mobile, and also provided it with the GPS so that each data point is georeferenced. A typical electromagnetic induction sensor has a transmitter coil, which initiates the process by sending electromagnetic waves in the subsurface. And then it has one or two pairs of receiver coils, which get the electrical conductivity information back from the subsurface. These receiver coils are either arranged horizontally to the transmitter coil, or they are arranged perpendicular to the transmitter coil. So this horizontal and vertical geometry determines the depth to which these receivers are going to receive the information, which is called depth of exploration. Our sensor has two perpendicular coils, which can measure up to the depth of 0.3 meter and 0.5 meter in the soil, and two horizontal coils, which can measure up to the depth of 0.8 meter and 1.6 meter. So in the field, we will tie this setup behind a ute and drive around across the whole field at a constant speed. So the methodology involves the EMI surveys, which we done on both sides in 2019. Two surveys were done in the dry season, and two surveys were done after the rains on both of these sites. The first step of getting the survey and then analyzing the data is to do inversion of the electrical conductivity survey. What is inversion and why we do it is, if this is a vertical soil patch in the soil, and we take the soil survey data from the field, our 𝒂𝒏𝒏𝒄𝒊𝒏𝒄𝒆 will give us information from 0 to 50 centimeter in the soil, 0 to 80 centimeter in the soil, and then 0 to 160 centimeter in the soil. So in this way, if we have information from the last soil array, we will not know that if this information is from this part of the soil or this or that. So mathematically, we split the data through inversion process. So after passing through the inversion, we get the information up to 0 to 50 centimeter, and then 50 to 80 centimeter, and then 80 to 160 centimeter. After this inversion process, we develop soil electrical conductivity maps. In the second stage, we took the soil samples from the field up to the depth of 1 meter, and then divided them into 3 soil depths. These soil samples were used to develop lab-based electrical resistivity to conductivity soil water calibrations, and also for doing all the soil physical and chemical analysis. In the third phase, we did the drone imaging for tree survival and tree crown growth detection. This drone imaging was passed through a stitching process of images, which is called orthophoto creation. And the next step, a canopy height model was developed. And in the next step, a machine learning algorithm was trained to detect the tree count and the ground size. And as a last step, we will validate these drone results with the ground truth data that we will get manually from the field. Once we have all this information, we will combine them to find the correlation of tree survival and crown growth with respect to ECM maps and soil parameters. Soil survey and sampling. This is our soil survey from the site of Kupring. The green dots are representing our soil survey transects, and the red dots are representing our soil samples points. Similarly, this is our soil survey from the 𝒏𝒗𝒊𝒓𝒐𝒏𝒏 site. And also we took the soil samples by using percussion drills up to the depth of 1 meter, and 22 soil cores were taken, which were developed into 66 samples. These are the first inverted result of our EMI survey. As you can see, our 𝒏𝒗𝒊𝒓𝒐𝒏𝒏 site was a uniformly textured soil. So there is a very low ECA conductivity was obtained in these maps, and there is not much difference across the depths as well. Also, when we took the survey of wet season after the rains, still the electrical conductivity of sandy soils is very low, but the survey shows that after the rains, the electrical conductivity has increased as compared to the dry season survey. Similarly, these are the inverted EMI survey maps for the site of Kupring. As the Kupring site has a variable textured soil, so that is shown in the inversion results. And similarly, just like 𝒏𝒗𝒊𝒓𝒐𝒏𝒏, after the rains, the ECA distribution has changed in the wet season survey. In the second step, we did the soil parameter analysis. The first one was determining the soil water, which we did by electrical resistivity to electrical conductivity power law calibrations. For this purpose, we took a known volume of soil sample and then saturated it, and then we measured the resistivity of this drying sample. This resistivity value is then used to calculate the volumetric water content. Since resistivity is an inverse of electrical conductivity, so this simple equation was used to spatially predict the soil water's status. This is the soil water map of 𝒏𝒗𝒊𝒓𝒐𝒏𝒏 site using this calibration for the dry season. And this is the soil water map of 𝒏𝒗𝒊𝒓𝒐𝒏𝒏 site for the wet season. Along with soil water, we did all kind of soil chemical and physical analysis for this purpose. The last phase was doing the drone imaging for plant survival and plant growth. So this part was outsourced through a company, 𝒂𝒄𝒂, which is based in the UWA. These were the Leptospermum plants at that time, where the drones were used to capture the images, which are very small plants at that time. And you can see that these plants at the same site has different growth rate. So this is very small, and this is very large, comparatively bigger plant. And this is a fully grown plant at that site during that time. So for the drone imaging, the first step was, like for the 𝒏𝒗𝒊𝒓𝒐𝒏𝒏 site, at least more than 500 drone images were stitched together for creating this orthophoto. And then a canopy height model was developed, which is a 3D point cloud data information. And then based on this data, a machine learning algorithm based on YOLO was trained. And using this algorithm, automated results were obtained to detect the trees in the field, to analyze the survival rate of the plants in these new sites, and also to measure the crown size of these fields. As you can see, that the larger circles are showing like bigger crown size or better growth of the plant as compared to the smaller size. At the end, the result, we have to do the validation, but currently, based on current available field ground truthing data, there is no correlation with the machine learning result in our results. So this could be probably, as you can see, a fully grown Leptospermum plant. I took the crown information by measuring the diameter at this height. But we really need to see that what did the machine learning took as a crown? Either they just only measured the top, or they have considered a certain height of that. So we are going to get the data again from the field, and also again looking into the machine learning algorithm, that what is actually considered as a crown in there. And then we will do this for validation again. So in the end, we have now soil ECM maps and our soil parameters information. We will be filling all those soil polygons with this soil information by doing some statistical analysis. And after that, once we have validated our drone information of growth rate and the crown size of these plants, we will be comparing that whether the growth rate or the survival rate of these plants in different polygons are different because of these soil parameters or not. So this is under analysis right now. So at the end of the result, we will be able to give some information to the farmers that what kind of soil are suitable for their leptospermum production. So far, 2 different soil sites are selected to evaluate the commercial production of leptospermum plants. One was 𝒏𝒗𝒊𝒓𝒐𝒏𝒏𝒎𝒄𝒊, which is a uniformly sandy soil, and the second one is variable texture open site. A mobile, relatively quick and inexpensive EMI survey is used to map the soil variations, including soil water and soil physical and chemical parameters. A drone survey and machine learning is used to determine the survival and growth rates of leptospermum plants. Correlating the variation of survival and growth rate with the soil variation will result in giving some information to the farmers about the suitable soil condition to commercially grow leptospermum. And in the end, ultimately, there will be an evaluation for the flowering and nectar production of these plants, which will be correlated to the soil sites, and we can develop some recommendations to the farmers for commercial production of leptospermum species. Thank you.
Thanks very much. Have you got any questions?
Either everybody understood or nobody understood.
No, it was very good. Yes, yes.
Was that a commercial leptospermum site that you were at? Is that somewhere where they're growing it commercially?
They are growing like for the very first time in Western Australia. So that's how we like studying those sites. So it's not like generally commercially grown.
But was that site that you did your survey on,
Yeah.
was that a commercial
Yeah,
site?
that was our commercial site, yes.
Yeah.
What do you refer by survival?
So the survival depends, like these plants in the same fields, they are newly planted. So at some sites they have died, and at some sites they just keep on growing. So we'll be analyzing that why some of the plants, newly planted plants, are like dying, and the other in the same field they are growing like flourishing. So
And I haven't seen the actual sites, but I think like the one site is heavier soil site, so you don't know. And it was very, very boggy. I think when Hira went in there, it got stuck, I don't know how many times. But you know, there could be water tables coming up high, and that's where the EM surveys might help, where you can pick up soil water levels and try and assess what's happening underground, you know, particularly with the soil water.
So if you see this map, so these plants were planted at the 6 meter distance all across the field. But you can see the empty patches here. So plants didn't survive in these parts. But at the same time, there are bigger plants. So that's fine.
Yes.
Here, is this plant susceptible to any diseases or insects or any pests that?
Not that we know, because this is just a native species. It was never grown commercially right now. So.
And are you also looking at a biotic biota in the rhizosphere of these plants, or talking with someone, or is someone looking at it in the project?
Because this is a part of honeybee program, so they have a lot of related projects, and this is my part is just with the soil and their two aspects. But yes, there are other groups which are working with them.
Yeah. So I think just So just to give a bit of background to Hira's overall PhD, she's looking at the use of EM induction to measure soil water. That's her overall objective. So she's got 3 major projects. One she's already published from Cunderdin in an ag environment. And then she's got another study at Litchfield. And then this is the 3rd study. So it's all related across the environmental and ag side, but really looking at the use of EM induction to measure soil water as a rapid method that might be useful for farmers and in this case for leptospermum.
Maybe I can add as well for the general public here. Farmers target marginal land to plant these leptospermum plants where normal agriculture practice did not turn out to be viable financially. Because these leptospermum plants seem to be very well adapted to harsh conditions, also to drought resistance and all that. That's why they select this marginal land to get potentially an additional income.
That was really nice talk, thank you. I'm curious about the, I guess, success rate of machine learning at actually detecting tree crowns, height and size, but also what kind of spatial resolution it can actually detect individual crowns from. So if they're closer together, at what point do you start to lose that capability?
Yeah, so because that part of the work was outsourced, so I don't know much about it, so I just got the result. But the resolution they took was like 1.3 centimeters. So yeah, so I think close. Yeah,
It's very interesting.
If not, thanks very much, Hira. You did a fantastic talk. Really good. Well, thanks very much. That's all for tonight. Look forward to seeing you next week.